So, when the DNA molecule we are talking about is unzipped, the bases of ACTG will be left exposed on a single strand of DNA. DNA contains millions of base sequences, but we will just consider this simple sequence of 4 as an example. So, when DNA is unzipped and the two strands are separated, then an enzyme called RNA polymerase will make a copy of one of the exposed strands of DNA, called the template strand. But it will make a copy in a complementary form, called mRNA or messenger RNA. What happens is that free-floating nucleotides, similar to the components of DNA, are joined in a molecule called mRNA or messenger RNA. This mRNA molecule matches up with the exposed DNA strand, so that if the mRNA molecule is matching with the nucleotide base G for guanine, the mRNA will have a C in that place, since C always pairs with G. The only difference between DNA and mRNA is that mRNA uses a slightly different coding system. DNA uses ATCG for adenine, thymine, cytosine, and guanine, but mRNA uses U in the place of T. mRNA uses U for uracil instead of T for thymine, so that its coding system is AUCG instead of ATCG. That means that when mRNA matches up with the single strand of DNA, and when it matches with A or adenine, it won't have a T as in DNA, it will have a U for uracil. When the process of base pairing is completed, that is, when the messenger RNA has made a complementary copy of the DNA template strand, then the mRNA molecule will break away as the DNA strands rejoin. The mRNA will then leave the nucleus and enter the cytoplasm. When the mRNA has left the nucleus, a protein called a ribosome will come along. This protein will grab the mRNA molecule. It is here where we move from the process called DNA transcription to the process called DNA translation. When the ribosome has the mRNA molecule in its grasp, so to speak, another molecule approaches called tRNA or transfer RNA. This is an extremely important part. Attached to the top of the transfer RNA molecule is an amino acid. Each transfer RNA recognizes only one amino acid. That's shown at the top of this image. At the bottom of the transfer RNA are 3 bases. These 3 bases pair up with 3 bases on the mRNA transcript we just discussed. You can see that in the images. What happens is that when the mRNA strand has the base sequence AUG, adenine, uracil, guanine, then the transfer RNA with the complementary bases at the bottom, and the specific amino acid which links with those bases, will pair up with the appropriate base pairs on the mRNA. In this case, UAC on the tRNA will match with the sequence AUG on the mRNA, because A is complementary to U, etc. Since the tRNA molecule has only 3 bases, as we see here, the coding or matching that it does in DNA translation occurs in sets of 3, as we see in these images. 3 base sequences, therefore, wind up expressing one amino acid. The 3 bases that do this on the mRNA, shown on the bottom, are called a codon. The 3 bases on the tRNA that pair with the codon on the mRNA are called an anticodon. In the example we're looking at, the codon would be AUG and the anticodon would be UAC. Once the first codon and anticodon pair up, then the mRNA slides along the ribosome to the next codon, and another tRNA molecule comes along with its specific amino acid at the top, and it matches the complementary bases to the next set of 3 bases on mRNA. The 2 amino acids at the top then become joined by a peptide bond, and the transfer RNA molecule is then released by the mRNA. The process continues until a chain of amino acids is formed. So what we have here is an actual code in which sets of 3 bases equate to or express a particular amino acid. There is a code and there is information.
Allow me to reintroduce myself. My name is Ho, H to the O-V. I used to move snowflakes by the OZ. I guess even back then you can call me CEO of the